freezers and the flies. Okay. Everyone in here now has a virgin male, female, except for John that I know of, right, John? Do you have a virgin female? You do not at all. You were originally white eye. Okay, so tomorrow you need to bring your flies. If not, I'll give you a virgin. But like, how about, does anybody, you've already killed your parents, right? Yeah. But your parents are dead, your parents are dead, right?
I don't have any.
Jack, are your parents dead? Or your parents alive? But what? Those. Okay. Do you have, that's what I'm asking you. Do you have pupa or worms?
Probably not.
Okay, do not kill your parents until you have pupa or worms. Presley, Savannah, sit. I can't multitask.
Sit down.
Okay, thank you. Where are your flies? Right where?
In my bag.
Okay, can you get them out for me? So I can see. Oh, it's so hard. It's okay. All right, now listen. Guys.
I mean, for my flies. Like forcing to come out. That's what I mean.
I'm okay with talking, but like you got to come back to me.
So.
All right. And what we've got, guys, I dealt with this last year or last semester a lot. I don't want to deal with this again. You guys are like slowly like going off this train of like, yeah, you can talk, but like as soon as I start talking, like just chill for a while. Got it? Okay, I don't want to have, I already had to give a seating chart to fifth period. I do not want to give it to seventh period. Okay, right. No, I mean like you pick these seats.
Yeah.
Okay. Yeah. Right. I pick the seats. Yeah. I don't want to do that, but if you can't shut up, then I'm gonna have to do it. All right, now listen. Okay, so what we've got is I've got these flies in the freezer. I'm going to give those to the people who need to do that. Okay, I want to remind you that all of you have this document. Okay, so today, Savannah, Isaac, Isabel, you need to pull this document up right now because you're going to be counting your flies. This will be day 2 counting for Savannah. Okay, everyone else is not counted yet. Okay, you do not want, at this point, nobody has to worry about their virgin female at all. Okay, all you have to do is worry about counting your offspring once you have offspring. Okay, we'll put the day, whether or not you have red males, red females, white males, white females. Everyone in this room could have different results. Okay, at this point, Savannah is the only one that had results, and this is on Friday.
What?
Oh, he had one. That's right, he had one. So today he had a red. Yeah. So he had one fly. I forgot about that. Isaac had. Savannah though had all white males, correct, and red females. Okay, we're gonna see if Savannah's got the same ones today or not. Question? Oh yeah. Okay, so now we're gonna watch a quick little video. We're gonna talk about protein synthesis. I'm gonna let the flies chill a little bit more. I'll get those in Petri. You guys got your Petri dish? You have a Petri dish? That's okay. I can give you a new one. You got your Petri dish? Okay. I'll give you that in a second. I'll give you some ice packs here too. Okay, now listen. Shh. I want you to watch this because we're gonna have a quiz on Wednesday. Okay, and I'll give you all the answers to the quiz, but I want you to watch this video first because hopefully this is gonna start to bring about some big picture kind of stuff. Okay. I love the
If you stay curious.
It could be worse, right?
I remember this video.
Well, it could be this particular one because we watched it. Okay.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information it codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA, well, a few exceptions, and that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine.
Are you noticing something different here? What's weird?
There's no T.
There's no T, right?
Yeah,
There's a
what's
U.
that?
We'll talk about
Me.
that.
A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA. So the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters. I remind you to stay curious.
Okay. Let me give you the answers for the quiz on Wednesday, right?
Yeah.
And you guys, Isaac, Savannah, Isabel, you guys can start counting. Okay. And then we can talk about that and you can pay attention to. Okay, you already finished? Okay, great. Okay, all right, real quickly. Here's the questions. Here's the answers. Ready? Okay, the first question will be this. If I have a DNA sample and the DNA is ATTCCG, what's the other side? Now, listen, it may not be that sequence, okay, but it'll be close to that. Y'all with me on that? Everybody can do that. If DNA is a double helix, one end is longer than the other one. What's this end that's longer? 5 prime, 3 prime. This one's what? 3 prime, this one's 5 prime, right? DNA always grows from blank to blank. What do I put there? DNA always grows from blank to blank. 5 to 3, right? Okay, 5 prime to 3 prime. We call that DNA replication. Okay, and don't forget, these little guys help unzip the DNA. Enzymes, right? Those enzymes do that. Okay, now that's the first. Let's see, that's question number 1, question number 2, question number 3. Okay, here's the next ones.
Yeah, I don't know the dude.
Who are these dudes? Who's this guy? Francis Crick. His partner's name was James Watson, right? This guy gave some information to, yeah, that's Morris Wilkins. He gave it to James Watson. He worked with this person. Got her. Okay, see ya. Okay, got it. Bye-bye. And then we got this guy. This is Shargat. He's the one that said the A's go with T's and C's go with G's. Guys, I might have to separate you guys. Seriously. Like, move up. Thank you. All right, last but not least, I got these two guys. This is Chuck Darwin, right, and Al Wallace. Okay, now here's how this is going to go down. I took selfies with these pictures in front of my face. Okay, I'm going to put them into a Google form. Okay, they will be kind of multiple choice, except here's the deal. I'm taking all those names, okay, and I'm putting them with each picture. So this picture, okay, will have the picture of me like this. Okay, got it? And then underneath it, you will choose the correct name. Here's your choices. Okay, Alfred Wallace, Charles Darwin, Francis Crick, James Watson, Rosalind Franklin, Irwin Chargaff, okay, and Morris Wilkins, right? Each 7 people will be on each picture. Are you all with me on that? Okay, this is going to be Wednesday. Okay, so make sure, and again, if you're like, I want to practice these people, how do I practice? All of these videos are on my YouTube. Okay, all you have to do is go to my YouTube. You can look at all these, or you can ask me personally, but there's no Quizlet for this. Okay, just pay attention in class. And there you go. You can make one if you want to, right? Okay, there you go. Okay, so there's that. Now, okay, there's my DNA. There's my opposite DNA. We know it grows from 5 to 3. DNA lives in what part of the cell? The nucleus, right? Okay, so remember, here's my nucleus. Here's my mitochondria. Here's my rough ER. Those ribosomes that are really, really important. Now, today, you learned that there's DNA, but there's also something called mRNA. Now, mRNA is really important. Does anybody know why RNA is like really in the news like lately? Like what's going on with RNA? What about COVID? Nope, nice guess. But there's something to do with the RNA and COVID. We'll talk about it in a minute. Okay, now here's the deal. Let's see if you can get this. If this is DNA, okay, what's the opposite RNA of this DNA? What would I have? Nope. A.
U.
A, U, U, G. Yep. We replace it. There's no T's. There's only U's. U stands for urine. So if you have to pee a lot, that's because you're producing. Oh, no, just kidding. Yeah. It stands for your. Did you get all your stuff?
Yeah.
Okay, cool. Want me to take this for you? Okay, what was your results?
Well, I have 21 white male and 12 white.
Whoa.
Count. You don't have any.
Can you go throw those outside?
Roll them.
You can just go right outside the store if you want.
Wait.
Are you done, Isaac?
They're away.
Huh?
They're away.
You do this. Go to sleep. No.
Wait, so her parents are the opposite of what hers.
Yeah.
Yeah,
Yo,
that's what they are.
you took them off the ice too long.
Because,
We'll see if she's right, right?
Go back to
because,
sleep.
yeah.
Okay, now I'm gonna give you all something.
They're raging.
Do not lose this piece of
I
paper.
think they're falling asleep.
won't lose it. Oh my God.
Okay?
I already lost it. I already lost it.
They're falling asleep. Quick, quick, quick. Come on.
There's one in there. There's one in there. I didn't get in there.
Well, here I can tell you. It's the red-eyed female.
Amino acid. Sammy sounds like a girl. They speak fast now.
I'll go.
No.
Get up. I'll just tell you when we're walking. Oh. I think we're gonna see.
All right, now what else? Let's do it. Let me go.
No,
Let's count it.
Then walking. No, ow! Yeah. Not in the classroom. Oh my gosh. Idiot. Oh, I thought you jumped out. Oh my gosh. No.
I don't know.
I put a plastic No! No! Keep breaking it.
Are you done?
Yeah.
Did you get his results?
I'm supposed to let this go now?
Put the lid on. Put the lid on. Put the lid on.
Are you good?
What are you doing?
All righty.
Flashbacks.
Flashback
Put
Friday.
it on.
You want me to let
I'll take.
You want them? There you go.
What were your results, Isaac?
Like 4, 3, and 4. Wait, 8, 8, 4,
What?
3.
What?
4 red and that's the 1 light blue.
Wait. Are you sure?
No,
What's going on?
not sure at all.
Oh. Are you sure you got white males and white females? Let's look and see if those are really white.
Yeah, Sammy, can we use your feet from?
Wait,
Oh
Yeah,
my
you just
god.
look at them.
I see 0 white females.
Okay.
What planet are you on, man?
I didn't count those ones.
No.
I counted the red ones. I saw 4 red ones.
Then
So that
why did
means
you
there's
put
like-
white females down on your thing?
If there's 4 red ones, that means the rest are females. That means they're white, right?
No. He's red-eyed hair.
And also there's one in
Listen.
here.
You gotta count what you see.
I counted the
Okay,
8.
look, here's what I'm counting right now.
Thing.
I see 0 white females.
Well, I
You
got
wrote
that now.
white females down.
Yes.
It's red female.
Red.
Red-eyed females.
Well, that's nice to know, guys.
I literally told you.
Don't listen to anybody but yourself. Count these flies,
Count
okay?
them.
He's the one that
You're
told
right.
himself it
You're
was
right.
red.
She is right, but like,
And we got one in here.
don't worry about it, okay?
Fine, that's dead.
Okay, everybody listen. to me. Shh, shh. Guys,
Listen.
guys.
I'm not telling you this to you. You're the one that-
You need to listen.
I'm gonna start taking phones away. Sam, I need you to close your Chromebook from now on. Once you get in here, it's off. Got it? You too. If I see that computer, I'm taking it. Okay? I know. No more. Okay? My patience is thin. Y'all with me on this? I'm taking phones from now on. Taking computers. No more computer. This is how it's gonna be. Okay? Trying to be cool, but I'm losing it. All of you in this room, when you get your flies, you need to count what you see. Okay? Please ask me if you have any questions. But when I look at his results, you have white females, white. I'm literally looking at his flies and I see 0 white females. All I see is white males and red females. Okay? Is that going to be everyone's results in here? No. Okay? you may get the right results. These might be right, okay? Tomorrow you might get a white-eyed female, okay? But I wanted to check. In fact, I would appreciate before you start like recording your stuff or after you record it, I'll check it to just back you up, okay? I have no problem with that, but refresh your male-female thing, right? Just so you know, okay, where you're at, okay? So here we go. He's got white males, red females. Okay, same with you. You both have the same thing, right? Correct? Is that what you had today? Okay, who knows what they're going to get tomorrow? Might get the same thing, might not get the same. Okay, all right. Now, at your tables, all of you have either a DNA, an mRNA, a tRNA, or an amino acid. Now, if you notice up here, I have ATT, then the mRNA is AUU, okay? But what about the tRNA? In this case, the tRNA is the opposite of the mRNA. So in this case, it would be UAA because RNA does not have a T. Okay? Yes. Oh, I didn't choose the right example. I mean, like, you know, I messed it up. So, okay, now look, here's what I got. She's got ATT.
Yeah.
Okay? Right? ATT and UAA match. Y'all with me on this? Okay, now this UAA matches this AUU. Okay, just like this. Okay, now we're going to be moving around a lot, not today, but later. If you are DNA, hold up your thing. DNA, my DNA people, DNA. Okay, look around. Here's your DNAs. Okay, if you're a DNA, you're on like a little train. You're a line of DNA. Okay, mRNA. Hold up your papers. You are a line of people. Okay. Now my tRNA. Okay, you are going to be matched up with an amino acid. You two are going to be together. Okay, right? You two are going to be together. You two are going to be together. Okay, you're a tRNA. Okay. I'm sorry. You and Isabel. You and Isabel. You and Oliver. Okay. And you and Chandler. Okay. Now here's the deal. Okay. tRNA and amino acid. You guys are pairs, okay? It's like you're stuck together and you're going to be moving around, okay? DNA, you're just a line, okay? In fact, you've got the easiest job ever because your job is to stay inside the nucleus of the DNA, or the DNA stays inside the nucleus of the cell, okay? It does not move whatsoever.
It doesn't.
Do you need me?
I need keys to the van. I'm gonna have somebody get that coat out before the school day is over.
Here you go.
Thank you.
You're welcome. Okay, you all with me on this? Okay, so DNA folks, okay, you're just gonna be one line. Okay, now in the past, we did like a little like choo-choo train kind of thing. We're not gonna do that this year because I don't want you to touch each other and give each other the sickness. Okay, right? mRNA, you'd be the little choo-choo train too. Okay, tRNA, you'd hold hands and jump around together. Okay, okay, we don't want to do that. We don't want to mix the spits and stuff. Okay, all right. Now listen though, no one answered the question. Why is mRNA important for COVID? I want to show you why and why more people should know about this wonderful thing called mRNA. Here's
How
why.
does an mRNA vaccine work? mRNA vaccines can be manufactured faster and more cost effectively than conventional protein-based vaccines. But how do they work? To develop a coronavirus vaccine, researchers selected an mRNA that encodes the coronavirus spike protein. This protein helps the virus enter a human cell. First, the mRNA is prepared by in vitro transcription or chemical synthesis. Next, the mRNA is encapsulated with lipid nanoparticles. This layer protects the mRNA and raises a stronger immune response. The final vaccine formulation is injected into the skin or muscle. Once the mRNA enters the cell, it uses the cell's translation machinery to produce the viral spike protein. The spike protein triggers the body's immune response to produce antibodies and specialized T cells. During future exposure to the coronavirus, these antibodies and T cells will attack the virus, preventing the development of COVID-19.
Okay,
How does an
so
mRNA...
typical vaccines like the ones you got when you were a little kid, those are protein based, okay? But ultimately, we will be making proteins. We'll talk about that on Monday. Or I mean on Tuesday, sorry. But today's Monday, right? Okay, thank you for your attention in. Riley, I will give, don't lose these papers. Take these papers with you. Bring them tomorrow, okay? Riley, I'm gonna make you DNA when you get back, okay?
Okay.
All right, peace out.